immediately. Thank you very much. We start immediately after the technical glitch. I want to thank the technical staff over there for solving these technical problems. So today, what we are going to do is that we are going to do a kind of summary or revision on the 3 topics we have taken. Remember, we have taken plant cell and living things. After living things, we took on the cells. After the cells, we now took on cell division. After cell division, we took on the viruses. So I want to, these 4 topics we handled, I want to kind of do a recap because of the time problem. Next time, we are going to start a new topic on the fungi. So today, let's summarize what we have done over the past 4 sessions we had. So the first thing we are going to look at now is we are going to start from where we start. I started in case people who do not follow us. We started from living things and non-living things. We summarize them, and then we say that those living things are those things that have life in them, and non-living things are those that do not have life in them. At the end of that lecture, I gave you assignments to look at 10 differences between living things and non-living things. And then, of course, we also did the characteristics of living things. These are the things we did in the first lecture. Remember, today I said we are going to summarize on the topics we have handled earlier. So after that, we now went to look at the cells. And then we discussed cells in details. And then we saw that every living thing is made up of cells. Whether the living thing is a plant or animal, we say that they have cells in them. And so that's what we summarized about cell. Then we went forward to look at the scientists that worked on this cell. Remember that after the discovery of microscope by Anthony Leeuwenhoek, a lot of research began to go on. And so some of these scientists now helped and then propounded what we call the cell theory. They propounded the cell theory, which we did last time. This is a kind of summary of what we have done before. And we say that cell theory states that all living things are made up of cell, and the cell is the unit of structure and function of life. And then we also said that that cell comes from pre-existing cell. These are the 3 cell theories, which we did last time. I just want you to recap, to have a knowledge of what we did before, so that in the next lecture that we're going to handle, it will be just a walkthrough. Then we also went forward to look at the 5 scientists that contributed to the development of cell theory. We talked about Robert Hooke. We talked of Matthias Schleiden. We talked of Felix Dujardin. We talked of Theodor Schwann. And we talked of Rudolf von Virchow. These are what we did last time. So, having looked at the cell theory and then the scientists that developed these cell theories, we now went to look at the structure of the cell. We drew a plant cell, we drew an animal cell. We labeled plant cell, we labeled animal cells. That's what we did last time. We also looked at the labeling of the plant cell, labeling of the animal cells. One thing that we understood was that this plant cell is large, while animal cell is smaller. And plant cells has cellulose cell wall. Animal cell do not have cellulose cell wall. Plant cell has a middle lamella. Animal cell do not have a middle lamella. Plant cell has very large vacuum. Animal cell has small vacuum, etc. Plant cell has the chloroplast. The animal cell don't have it. So that's what we discussed. And then I gave you an assignment to list 10 differences between plant cell and animal cells. And then we also looked at the similarities. I asked you to list 5 similarities. Both of them have cell membrane. Both of them have nucleus. Both have cytoplasm. Both have mitochondria. Both have cell membrane. And then both have nucleus. This is what we discussed last time. Then from there, we now went to cell division. I just want a recap of what we did last time. We went and did the cell division. And then we said that there are two types of cell division. That's mitosis and meiosis. We said that for any living organism to grow, for any living organism to continue to live, the cells must continue to divide so that the organism will either increase in size or length. So, and that's why division is very, very necessary in any living organism. If you plant a maize, maybe maize seed, you expect that maize seed to germinate. And when it germinates, you expect it to grow and produce fruits. That is division taking place. We also, in animals, when a child is born, we don't expect the child to continue to be small as the day he was born. We expect the changes to take place, and that division that makes it possible. So we say whether we are talking of mitosis or meiosis, that of course, you know that mitosis takes place in this body cells or somatic cells, while meiosis takes place in the reproductive cell. We say whether we are talking of mitosis or meiosis, that there's stages of the division, even though in the natural sense, they are not physically divided. They occur simultaneously. But what we did is to break them into stages so that it will be easy for us to understand. And that's why we looked at stages of cell division. We say the interphase, the prophase, the metaphase, the anaphase, and telophase. These are the stages of cell division. Now, we also went forward to look at the mitosis. I used a cell. I gave you an example of a cell that has 4 chromosomes. I say with the use of labeled drawings, describe the process of mitosis in this cell. That's the assignment I gave you last time. So this is just a recap of what we did earlier. Today's lecture is a kind of recap. We want to bring us together in all the lectures the 4 lectures that we have had, so that in the subsequent lectures, we'll continue from where we stopped. Now, we say for that cell that has 4 chromosomes, we say what happened at the interphase? At the interphase, the chromosomes are not well distinct. They are coiled together. Then at early prophase, it is possible for you to see the chromosomes. Now, at late prophase, the chromosomes have contracted and very distinct now. You can observe them. And then the centroids now begin to form. And the nuclear membrane will begin to disintegrate. And then when we come to the metaphase, the early metaphase, the nuclear membrane has disintegrated. And then the centroids will begin to migrate to the opposite pole. And then the chromosomes at this time will now begin to move to the center of the cell. So at late metaphase, all the chromosomes will come align themselves at the equator of the cell, at the center of the cell. Each and every one of them occupying one spindle fiber. So the spindle fiber will hold them at the center of the cell. Then at early prophase, something will happen. Remember that a chromosome is made up of daughter chromatids held by the centromere. So at the early prophase, what happens is that these daughter chromatids will now begin to separate, and the spindle fiber will begin to pull them apart. So they begin to separate. At late anaphase, now they have separated and they start migrating to the opposite pole. Now, the next one is what? Telophase. At early telophase, now you see something like a constriction trying to appear. Of course, the daughter chromatids have migrated to the opposite poles. And then constriction will now begin to appear, showing you that division is about taking place. Now, at late telophase, division actually takes place, and the cell now divides into 2. So one cell at the end of mitotic division divides and gives rise to 2 daughter cells. But each of these 2 daughter cells formed have the same chromosome as the parent cell. That's the one thing we notice at mitosis, that the daughter cell will divide and give rise to two daughter cells. It is very, very pertinent that we notice this thing very here. One cell divide and give rise to two daughter cells. Now, we now go to and then each of the daughter cells has the same number of chromosomes as the parent cell. You have to know this. Because when we come to meiosis, you see a different thing happening. Then we'll come to meiosis. Now, listen very well, because it's a technical thing of which you must understand it as somebody who has passed through this biology class. Now, meiosis takes place in the sex cell or the reproductive cell. And that is the what? In the sperm and in the what? In the ovary. This is where it takes place to produce the sperm and to produce what? The ovary or the ovum. Now, what happens is that let's use that cell that has 4 chromosomes. Let us see the stages of meiosis in this cell that has 4 chromosomes. Now, in meiosis takes place in 2 stages. Mitosis did not take place in 2 stages, but meiosis takes place in 2 stages. You have what to call the first meiotic division and second meiotic division. Let's look at what happened at the first meiotic division. At the first meiotic division, what happened is that at the early prophase or at the interphase, the chromosomes are not very distinct. It cannot be distinguished. Now, at this early prophase 2, the early prophase 1, the chromosomes begin to be distinct, and you can see them. At early prophase 2, what happens is that the nuclear membrane, early prophase 1, the nuclear membrane will begin to disintegrate. Then when we now come to the metaphase, early metaphase 1 and late metaphase 1, the chromosomes at the early metaphase are going to the center of the cell. At late metaphase 1, they have reached the center of the cell and they will align themselves at the center of the cell. But then it is no longer one chromosome to one spindle fiber. This time around, they will pair up. They will be in pairs. So 2 chromosomes will pair up at the center of the cell. They don't go single, they go in pairs. That's why we say that daughter homologous chromosomes pair up at the equator of the cell. That's what we discussed last week. This is just a revision to take us back to what we did and to make sure that all of you followed. Even though you did the assignment, I read the assignment and saw that some of you just copied the work of others. So how can I score you? If you know you did that, then you are into a very serious problem. So let's leave metaphase and go to early telophase 1 and late telophase 1. At early telophase 1, there is a constriction that is appearing. There is a constriction that is appearing so that you will now see that, that the chromosome is about dividing. You will now see that the chromosome is about dividing, that the chromosome will divide. So the cell will divide into 2. So when they divide, it gives you to 2 daughter cells. Now, the chromatids of this chromosome have not yet separated. That's why the 2 chromosomes formed will still enter into the second meiotic division. So the first chromosome, the first cell will undergo second meiotic division. The second cell will also undergo second meiotic division. That's what happened now. Now, let's see what happened now. Let's take the first cell, for example. The first cell will enter early prophase 2 and late prophase 2. At early prophase 2, the chromosomes are now very distinct, and then you can see them very well. At late prophase 2, the nuclear membrane will begin to disintegrate, and the central will begin to move to the opposite pole. Now, at early metaphase 1, the chromosomes will now begin to come to the center of the cell. At late metaphase 1, it has come to the center of the cell. This time around, they are no longer pairing. They paired at the first meiotic division. At the second meiotic division, they are not pairing. What is happening here is that the daughter chromatids will now separate, because the essence of pairing in the first meiotic division is for crossing over to takes place. Crossing over, that's the mixing of the chromosomes. They take place in the first meiotic division. In the second meiotic division, the daughter chromatids will separate. And remember that this daughter chromatids separating now have the information. They have the genetic material of both the mother and the father. So here at second, at metaphase 2, they align themselves at the equator of the cell, each of them occupying a single spindle fiber, no longer double staying in phase as we find in metaphase 1. In metaphase 2 is one chromosome to one spindle fiber. So they will now stay there. And the early anaphase 1, the chromatids of these chromosomes will now separate. At late anaphase 1, they have reached the opposite pole. Then you now come to the telophase. At the early telophase, the constriction will now occur, showing you that division is about taking place. At late telophase, it divides. Now, the first cell divides to give rise to two daughter cells. The second cell divides to give rise to two daughter cells. So, in the end of meiosis, 4 daughter cells are formed. And when you look closely at these 4 daughter cells that form, each of them has half of the number of chromosomes as in the parent. So each of the daughter cells formed has half the number of chromosomes as the parent. So the parent, as this example we use, has 4 chromosomes. Each of the daughter cells now has two chromosomes. So the summary is that at the end of meiosis, 4 daughter cells are formed, and each of these daughter cells has the number of chromosomes as the parent cell. That is meiosis for us. Recall what we did at mitosis. That at the end of mitosis, how many daughter cells are formed? 2. How many chromosomes are in each of the 2? The same number of chromosomes as the parent cell. Now, look at meiosis. How many daughter cells are formed? 4. How many chromosomes are present in each of these daughter cells? Half the number of chromosomes as in the parent cell. Let this thing get into your head. It's a kind of summary of what we did the last sessions that we have not able to hold. So from this, we have seen the summary. We have summarized everything. That at the end of mitosis, 2 daughter cells are formed. And each of the daughter cells has the same number of chromosomes as the parent cell. Then at the end of meiosis, 4 daughter cells are formed. And each of these 4 daughter cells formed has half number of chromosomes as the parent cell. That is the summary of mitosis. Then the question are, mitosis, are they important? Meiosis, are they important? Yes. The importance of mitosis. Now, advantages of mitosis. I said that it helps in the growth of an individual. For you to grow and reach this level you have, mitosis is taking place. If you have a cut or you have an accident, after the doctor must have done an operation, your body will heal. What is happening in this healing? Mitosis. So it helps in the growth of dead tissues or replacement of dead tissues. It helps in the replacement of dead tissues. It helps an organism to grow. That's the 2 things. 1, it helps an organism to grow. Then 2, it helps in the replacement of dead tissues. And then 3, it helps in the multiplication of cells. Because, for example, if bacteria enter your body, before you know it has multiplied, what is helping it to divide and divide and divide? It is what? Mitosis. Then also, it helps also to produce individual with the same gene, with the same genetic makeup as the parent. Because the mitosis, there is no mixing of chromosomes. So the individuals produced is a replica of the parent cell. So let's look at meiosis. What are the importance of meiosis? It helps in reproduction. It helps in reproduction because for you to reproduce, the number of chromosomes in the sperm have to be half. The number of chromosomes in the egg has to be half, so that when fertilization takes place, they return to the normal. So meiosis helps in reproduction. And also meiosis helps in the what? Mixture of chromosomes. Because the chromosomes inherited by the father also, and the one that the mother, the child inherits both of them. And now has the qualities of the father and the qualities of the mother. Now, let's look at, okay. I'm reading what you are saying. Yes, I've read that you are following the lecture. Thank you very much. Now, let's look at differences between mitosis and meiosis. I asked you to give 10 differences. Number one, under mitosis, it takes place in the body cells. Under meiosis, it takes place in the reproductive cells. Number two, mitosis results in the production of what? 2 daughter cells. Meiosis results in the production of 4 daughter cells. Under mitosis, each of the daughter cells has the same number of chromosomes with the parents. Then under meiosis, each of the daughter cells produced has half the number of chromosomes as the parent cell. Under mitosis, crossing over does not take place. Under meiosis, crossing over takes place. Under mitosis, homologous chromosomes do not pay up. Under meiosis, homologous chromosomes pay up. Under mitosis, only one stage of division takes place. Under meiosis, 2 stages of division take place. Now, from there, we want to look at cell cycle. This what I'm just repeating or recapping what we did in our previous lectures. So, with this cell cycle, and we say that cell cycle are a series of events that takes place in a cell as it grows and divides. As the cell grows and divides, there is stages that it takes place. And I told you last time to put 5 boxes. To put 5 boxes and label the boxes. The first one you label G1. So you have G1 phase. The second one you label S. That means S phase. The third one you label G2. And that is G2 phase. This G1, S, and G2 phase takes place at the interface. Remember, when we did cell division, I gave you stages, IPMAT, 5 stages: interphase, prophase, metaphase, anaphase, and telophase. So in the cell cycle, the first box, which is G1, the second box, which is S stage, and the third one, which is G2, 3 of them takes place in the interphase. So now the fourth box is mitosis, and the 5th box is cytosis. So, or cytokinesis. The 5th box is cytokinesis. So if you are asked to mention the stages of cell division, of cell cycle, 1 is the G1 stage, the S stage, the G2 stage, the mitosis, and then the cytokinesis. What happened at G1? I said that the G1 is the primary growth phase. And here, that is where this is where the growth of the cell takes place at the G1 and is the longest phase. Then the S phase is the synthesis phase. The synthesis phase, where the cell synthesizes a replica of its DNA. So DNA is synthesized in the S phase. And then the next one is G2 stage. What happens as G2 stage? So this is where the cell is preparing for division. So this is where the cell is involved in preparation for separation and replication of the cell genome. It prepares for the division. Then the next one is mitosis. Mitosis where the cell divides. And then the next one is cytokinesis. Of course, the cytokinesis is the stage in which the cell divides, creating 2 daughter cells. So, of course, you know that the mitosis is, remember, the mitosis involves interphase, prophase, metaphase, anaphase, and telophase. So the cytokinesis is where the cell divides into 2. So these are the stages of the cell cycle. From there, we want to look at viruses. From here, we went and looked at viruses. We are just repeating all the lectures we did. Those of us that we are joining for the first time, is an opportunity for you to catch up what we were able to do. Next lecture, we are going to start the fungi and then we move forward. And very, very fast. I just want to recap what we did because of the technical glitch that we have today. Now, viruses. We said that the viruses are microorganisms that can be seen with only electron microscope. You can see it with electron microscope. All area that you can see with optical microscope. And then, of course, we look at the characteristics of virus. He said cannot be seen with our naked eyes, can only be seen with the use of electron microscope. And they do not have they are very simple and do not have complex cell structure. They live inside the other organisms as parasites. So when you draw the structure of a virus, further you have the DNA inside, then it has a protein coat and a very simple structure, but then it's a very dangerous organism. Then we looked also at HIV, which is also a virus. Then we say that what happened? That this HIV virus, what it does is that it attaches itself to the receptor protein in the cell surface membrane. If the cell enters your body, it goes directly into your own cell and attaches itself in the protein, the receptor protein in the cell surface. And then as time goes on, it now enters the cell by endocytosis. And when it enters the cell by endocytosis, now the viral RNA is now released. They now release because it's now released into the cytoplasm of the host together with an enzyme called the transcriptose. Once he releases his own RNA, then with the help of this transcriptose, it now goes into your own cell and then now form a double-stranded DNA. So from a single-stranded RNA that has come into your distance, it now forms a double-stranded DNA. Now the DNA enters the nucleus and inserts itself into the host DNA. You can now imagine what happened. A foreigner that came into your body will now take possession of your own DNA. It enters the nucleus and attaches itself into your own DNA. Now, when it enters your host DNA, now whenever the cell divides, it also makes another copy of this viral DNA. When the cell now divides, give rise to another cell, the other copies that are coming now has what? This virus. The virus DNA is in that new cell that has formed. Now, after a very period of latency, maybe about 5 years, the virus became activated and now claims the ownership of the compound. It now claims the ownership of the house. It now claims the ownership of the nucleus and begin to damage and begin to destroy the original nucleus and take over the place. That's what we discussed in our previous lectures. I hope you followed our revision. Now, some of you have already submitted the assignment. Some have not submitted the assignment. And then some even copied. I looked through your assignment. I saw that some of you went and took other people's work and did. It's not good. If you know you did that one, cancel it and submit the original one because I'm going to withdraw all of you that copied from the other one. Now, when we come to the next lecture, we look at fungi. You see that what will happen is that fungi is a plural. The singular is what? Fungus. And then you have something like rhizopus, like mucor. All these are fungi. And then you see that they are not microscopic. You can see them with your naked eye. Of course, you know that under cell, you have what we call the prokaryotic cells. And then the other one that are not prokaryotic, the eukaryotic. So the fungi are under eukaryotes. And now what will happen? When they grow, they now release through their hypha, which they grow. For example, if you buy bread and keep it in your wardrobe for 1 week, when you now bring out that bread, you see some growth on top of it. That is the fungi, mucor, that is growing on your bread. And then they have what we call the hypha and the rhizos that goes into your bread. They secrete materials into that, and they now absorb the digested material into its body. So you now find out that fungi is a special group of organisms. Unlike our plants, they are heterotrophic because they cannot manufacture their own food. They feed on the digested food materials of the host, the dead and decaying material of the host, because they lack chlorophyll. So we are going to see in details. I think in our secondary school, we drew that. I remember mucomucedo, rhizopus nigricans. This is what we did in secondary school. And you can still recall your secondary school lecture, how you can, even the hypha, you can label it. And then the rhizos inside the host, you can label it. So these are what we are going to do in our next lecture. We'll do the fungi in detail, and then we'll now move forward and look at other things. I want to receive some of your questions. Ask your question, because this lecture is a kind of recap of all the things we have done before, so that in our subsequent lectures, we can now flow as one, because I know that some of you missed the lectures. And that's why I use today as a Father Christmas for all of us to recap all the things we know we have done before, so that we can move forward. I hope you are happy with the lecture. Thank you, and give us. I want you to ask some questions so that we can move.
Good evening, sir.
Yes.
Thank you for today's lecture. So
Yes.
I want to ask, how long does the mitosis and the meiosis process take
Now,
for it to come to conclusion?
Very good. I told you before that all these stages, apemat, the interphase, the prophase, the metaphase, the anaphase, and telophase occur simultaneously. So in terms of time, whether it is 5 seconds or whether it is 3 seconds or whether it is 1 hour, depends on the physiological processes, physiology of the organism that is concerned, but it takes place under a short period of time. So like now, if it is like 1 year for the cell to divide, you cannot grow to this level. I hope you understand. So the cell division and the growth of the cell depends on the individual. For those that are not feeding well, you see that they don't grow very well. But those that came from well-to-do family, you see how fast they grow, because they have all the enablement that makes the cell to divide and grow fast. So I need more questions. Thank you for that brilliant question.
Wow. Now I understand. That's analogy that you use about people from poor family and rich family is
Not
relatable.
rich per se, but well
Like
to
those that are
well
feeding well.
family.
Sir.
Continue. Yes, I can. Go on.
Good evening, sir.
Go on. I'm listening. Yes.
Yeah, thank you for your lecture so far. I just wanted to ask.
Yes.
Can you hear me? Hello?
Yes.
Can you hear me?
Yes, I can hear you.
Okay, please, I just want to ask.
I can hear you very well.
Can you please explain how cloning works with mitosis and meiosis? Cloning.
Yes. You remember that cloning is another biotechnology tool. Do you understand? And then cloning is the ability to make up a new individual. Do you understand?
Yes, sir.
And then cloning does not involve reproduction. Do you understand? And of course, you remember the first animal that was cloned. Remember the first animal, Dolly, was cloned.
Yes.
You can go to the laboratory and pick. What happened here is that you can pick a cell from a body cell, somatic cell, and then go into the lab and manipulate. The full technology cannot be explained here. But what happened here is that it does not involve the unification of the male sex cell and the female sex cell. All you need is to take a body cell from any of the organisms and then manipulate the cell in the laboratory to get a new organism.
So cloning has to do with body's mitosis, more of
no
mitosis.
reproduction. No reproduction. Yes, it has to do with mitosis.
Okay, sir.
I hope you understand. Thank you. And that's why in cloning, you produce a replica. You produce an exact copy. And that is the danger of cloning. So if you have a very evil man, evil person, and you claim that individual, the evil will continue to perpetuate. And of course, ethically, we don't allow cloning human.
Okay. Thank you very much.
Go on. Yeah, more questions. Titus, do you have a question? Okay, more questions. We need more questions. Interact with us. Science classroom is not an authority. Nobody is an authority in science lecture. So it is a kind of give and take, a kind of triangular method of teaching in science classroom. Interaction going on between the student and the lecturer, and then between the students and students, and then back to the lecturer again. So if you have any question, you have 4 more minutes to do that.
Sir, please, my question is in HIV now, for example, during the time that the virus keeps spreading in the body by means of the cells that are being replicated, is it possible to stop it? Is it possible to
Yes,
stop
it is.
to it?
What happened is that I'm not very sure of the drug, but I think they are saying that developing drugs that can suppress it. I hope you understand. But the best thing is not allow it to reach that stage, and that's why if somebody has somebody can take that, you go and start taking the drug as early as possible, so as to prevent this viral DNA from taking over your own DNA, your own nucleus. Because when they take over the nucleus, they now begin to multiply. Mitosis will now begin to take place and then replicate and give more. And when they spread all over the body, it becomes very difficult to control.
Okay,
More
sir.
questions?
Thank you.
Thank you.
Hello, sir.
Yes.
So please, I'm just confused about something a bit.
Titus, ask your question.
Yes, sir. Like you talk about HIV virus in human,
yes.
And then you said it attacked the cell. It goes straight into the cell and then attack. Then how comes that HIV patient will give birth to a child that is not HIV positive, sir?
Okay. You remember the reproduction. You remember there is a placenta.
Yes.
That supplies the
Yes,
mother's nutrients from the mother to the child. And then that's why if somebody need to go for antenatal. Once they discover it, it is possible to hold it so that the womb pass through the what? That placenta that is the connecting link between the mother and the child. Do you understand? So that the HIV, the virus do not migrate to that individual. And during childbirth, most important, so that during the childbirth, it will not migrate. So that's why one has to give himself, he or herself to medical distance so that they will be away, so that during birth, you can have a normal birth, and then they separate without mix up of the blood.
Okay, so it means there is a high risk of the child getting the HIV.
If it is not handled very well, but the child will not have the HIV until the child is born. So mostly it is during time birth that this contamination takes place. Do you understand?
Yes, thank you, sir. Yes, sir.
Thank you. We have 2 more minutes. 2 more minutes. Peter, do you have a question?
Thank you, sir. Also, I listen, sir.
So who is asking?
I said I want to ask a question based on the virus, sir.
Okay. Is it promise?
Yes, sir.
I'm going to ask.
Okay, so you said when virus is in a living organism, it behaves like a living thing, and when it's out, it behaves like a non-living thing, right? So,
okay. The whole thing is.
My question is, why is it?
Okay, very good. Thank you very much. That's virus is a very complex organism. That's why we should all keep away from contacting the virus, because their behavior is not they have complex behaviors. So that's what we have been able to handle today. I hope that this revision is very fine. We have been able to handle from the first lecture to this one. So the next one we move. Make sure you attend the next lecture. And when we come next week, I will come on time so that at 3 o'clock dot we start, and we have to be very, very fast like so that we can cover what is scheduled for outside us in the syllabus.
Sir,
I have enjoyed you very much.
My question is,
thank you very
why
much,
is it behaving like you not living outside?
That is virus for you. You cannot determine the behavior of virus. It's a very stubborn disease. Do you understand? So that's why you have to be very. It's not only HIV, other viruses. You remember when we have coronavirus? Did you find it easy? Every person was mad with coronavirus. That's it. We have other viruses that are around. So thank you, and God bless you for today's lecture. I've enjoyed all of you. God bless you, and we will end it here. Thanks.